What is the first intermediate produced in the Krebs cycle? A. Citrate. B. Isocitrate. C. Succinyl-CoA. D. Fumarate. The correct answer is A. Citrate is the first intermediate produced in the Krebs cycle and is formed from the condensation of acetyl-CoA and oxaloacetate. What is the most important purpose of the Krebs cycle in cellular respiration? A. To produce NAD and FADH2. B. To produce ATP. C. To break down glucose into pyruvate. D. To convert NAD to NAD. The correct answer is A. The Krebs cycle's most important purpose is to produce NAD and FADH2. These carry high-energy electrons to the electron transport chain for ATP production. Which enzyme converts citrate to isocitrate in the Krebs cycle? A. Aconitase. B. Citrate synthase. C. Isocitrate dehydrogenase. D. Succinyl-CoA synthetase. The correct answer is A. Aconitase is the enzyme responsible for converting citrate to isocitrate in the Krebs cycle. Which of the following is a citric acid cycle intermediate that can be used for gluconeogenesis? A. Citrate. B. Succinate. C. Oxaloacetate. D. Malate. The correct answer is C. Oxaloacetate is a citric acid cycle intermediate that can be used for gluconeogenesis, as it can be converted to phosphoenol pyruvate. In the Krebs cycle, which step makes GTP from succinyl-CoA? A. Citrate to isocitrate. B. Isocitrate to alpha-ketoglutarate. C. Alpha-ketoglutarate to succinyl-CoA. D. Succinyl-CoA to succinate. The correct answer is D. The conversion of succinyl-CoA to succinate involves GTP formation during the Krebs cycle. Which coenzymes are reduced during the Krebs cycle, leading to the production of NAD and FADH2? A. NAD and FADH2. B. FAD and FADH2. C. NAD and FAD. D. NADP and NADPH. The correct answer is B. FAD is reduced to FADH2, and NAD is reduced to NAD during the Krebs cycle, generating high-energy electron carriers. What is the final product of the Krebs cycle that combines with acetyl-CoA to start the cycle again? A. Oxaloacetate. B. Citrate. C. Succinate. D. Malate. The correct answer is A. Oxaloacetate is the final product of the Krebs cycle that combines with acetyl-CoA to start the cycle again. In the Krebs cycle, which enzyme converts succinyl-CoA to succinate? A. Citrate synthase. B. Isocitrate dehydrogenase. C. Succinyl-CoA synthetase. D. Succinate dehydrogenase. The correct answer is C. Succinyl-CoA is converted to succinate by the enzyme succinyl-CoA synthetase, which generates GTP during this process. Which of the following is an essential coenzyme for the Krebs cycle reactions? A. ATP. B. NAD+. C. ADP. D. FAD. The correct answer is B. NAD+ is an essential coenzyme that gets reduced to NAD during Krebs cycle reactions, helping in the transfer of electrons. Fumarate is converted to malate in the Krebs cycle by which enzyme? A. Aconitase. B. Citrate synthase. C. Fumase. D. Isocitrate dehydrogenase. The correct answer is C. Fumarate is converted to malate by the enzyme fumase, contributing to the continuation of the Krebs cycle. Which Krebs cycle intermediate is directly involved in the electron transport chain to generate ATP? A. Succinate. B. Malate. C. Fumarate. D. Oxaloacetate. The correct answer is A. Succinate is an intermediate oxidized to fumarate by succinate dehydrogenase, feeding electrons into the electron transport chain for ATP production. What is the significance of the Krebs cycle in cellular respiration beyond ATP production? A. Production of NAD. B. Generation of intermediates for biosynthesis. C. Oxidation of glucose to pyruvate. D. Release of carbon dioxide. The correct answer is B. The Krebs cycle generates intermediates essential for biosynthetic pathways, providing precursors for amino acids, nucleotides, and other molecules. What is the final product of the Krebs cycle before starting over with a new round of acetyl-CoA? A. Fumarate. B. Succinate. C. Malate. D. Oxaloacetate. The correct answer is D. Oxaloacetate is regenerated at the end of the Krebs cycle to combine with a new acetyl-CoA molecule, restarting the cycle. Which enzyme converts malate to oxaloacetate in the Krebs cycle? A. Aconitase. B. Malate dehydrogenase. C. Citrate synthase. D. Isocitrate dehydrogenase. The correct answer is B. Malate is converted back to oxaloacetate by the enzyme malate dehydrogenase, completing the Krebs cycle. Which enzyme converts alpha-ketoglutarate to succinyl-CoA in the Krebs cycle? A. Citrate synthase. B. Isocitrate dehydrogenase. C. Alpha-ketoglutarate dehydrogenase. D. Succinyl-CoA synthetase. The correct answer is C. The enzyme alpha-ketoglutarate dehydrogenase is responsible for converting alpha-ketoglutarate to succinyl-CoA in the Krebs cycle. What is the significance of Krebs cycle intermediates for biosynthesis of non-essential amino acids? A. Provide energy for amino acid synthesis. B. Supply carbon skeletons for amino acid synthesis. C. Act as cofactors in amino acid synthesis. D. Facilitate enzymatic reactions in amino acid synthesis. The correct answer is B. The Krebs cycle intermediates provide carbon skeletons for the biosynthesis of non-essential amino acids. Which Krebs cycle intermediate can be converted to oxaloacetate, used for gluconeogenesis in the liver? A. Malate. B. Succinate. C. Fumarate. D. Citrate. The correct answer is A. Malate. A Krebs cycle intermediate can be converted to oxaloacetate. Oxaloacetate is then used for gluconeogenesis in the liver. During the Krebs cycle, what is the role of succinate dehydrogenase? A. Convert succinate to fumarate and produce FADH2. B. Form isocitrate from citrate. C. Generate ATP from succinyl-CoA. D. Facilitate the conversion of oxaloacetate to citrate. The correct answer is A. Succinate dehydrogenase converts succinate to fumarate and produces FADH2 during the Krebs cycle. What is the common product of the oxidative decarboxylation reactions in the Krebs cycle? A. ATP. B. NAD. C. Carbon dioxide. D. GTP. The correct answer is C. The common product of the oxidative decarboxylation reactions in the Krebs cycle is carbon dioxide. What is the key enzyme responsible for converting isocitrate to alpha-ketoglutarate in the Krebs cycle? A. Isocitrate dehydrogenase. B. Citrate synthase. C. Alpha-ketoglutarate dehydrogenase. D. Fumase. The correct answer is A. Isocitrate dehydrogenase is the enzyme that catalyzes the conversion of isocitrate to alpha-ketoglutarate. This is a crucial step in the Krebs cycle. What is the immediate precursor molecule for citrate synthesis, combining with oxaloacetate in the Krebs cycle? A. acetyl-CoA, B. succinate, C. malate, D. fumarate. The correct answer is A. Acetyl-CoA is the precursor molecule that combines with oxaloacetate to form citrate in the first step of the Krebs cycle. Which Krebs cycle intermediate can be converted into oxaloacetate, a key molecule for gluconeogenesis in the liver? A. fumarate, B. malate, C. citrate, D. succinyl-CoA. The correct answer is B. Malate can be converted into oxaloacetate, an important precursor for gluconeogenesis in the liver during times of increased glucose demand. Which coenzyme in the Krebs cycle accepts electrons and becomes reduced to NAD and FADH2? A. NAD-JOR, B. ATP, C. NAD, D. FAD. The correct answer is A. NAD+ is a coenzyme that accepts electrons during the Krebs cycle, becoming reduced to NAD. NAD then carries electrons to the electron transport chain for ATP synthesis. What is the primary purpose of the Krebs cycle in cellular respiration, apart from energy production? A. To release carbon dioxide. B. To generate building blocks for biosynthesis. C. To produce water. D. To regenerate ATP. The correct answer is B. The Krebs cycle produces key intermediates. These intermediates are used as building blocks. They're used for biosynthesis of macromolecules. Macromolecules like lipids, amino acids, and nucleotides. How many acetyl-CoA molecules are generated from one glucose molecule to enter the Krebs cycle? A. 1 acetyl-CoA. B. 2 acetyl-CoA. C. 3 acetyl-CoA. D. 4 acetyl-CoA. The correct answer is B. Through glycolysis, one glucose molecule is converted into 2 pyruvate molecules. Each pyruvate then enters the Krebs cycle as 1 acetyl-CoA. This results in a total of 2 acetyl-CoA molecules per glucose molecule. Which Krebs cycle intermediate is directly involved in heme biosynthesis, a crucial part of hemoglobin? A. Alpha-ketoglutarate. B. succinyl-CoA. C. malate. D. citrate. The correct answer is B. Succinyl-CoA is directly involved in the biosynthesis of heme, an essential component of hemoglobin and other hemoproteins. What is the function of Krebs cycle intermediates in making non-essential amino acids? A. They provide energy for the process. B. They act as building blocks for amino acid synthesis. C. They regulate enzymes in the pathway. D. They contribute to cell membrane formation. The correct answer is B. Krebs cycle intermediates serve as precursors for the synthesis of non-essential amino acids, which are vital for protein production in the body. What is the significance of Krebs cycle intermediates for making neurotransmitters like serotonin and melatonin? A. They're used as energy sources for neurotransmitter production. B. They're converted directly into neurotransmitters. C. They act as precursor molecules for neurotransmitter synthesis. D. They regulate neurotransmitter release in the brain. The correct answer is C. Krebs cycle intermediates serve as precursor molecules for the synthesis of neurotransmitters like serotonin and melatonin. These are essential for neurotransmission and mood regulation in the body. Which coenzyme in the Krebs cycle accepts electrons and gets reduced to NAD and FADH2? A. ATP. B. Coenzyme A. C. NAD-JOR and FAD. D. GTP. The correct answer is C. NAD+ and FAD are coenzymes that play a crucial role in accepting electrons and becoming reduced to NAD and FADH2, carrying them to the electron transport chain for ATP production. What is the role of citric acid cycle intermediates in making non-essential amino acids? A. They provide energy for amino acid synthesis. B. They act as building blocks for some non-essential amino acids. C. They boost the activity of amino acid making enzymes. D. They change essential amino acids into non-essential ones. The correct answer is B. Citric acid cycle intermediates can act as precursors for the biosynthesis of non-essential amino acids in the body.